Have you ever wondered how every living being on Earth is uniquely coded? The answer lies in a molecule so fundamental, it's present in almost every cell of your body: DNA. Just like a blueprint for a house, DNA holds the instructions to build and maintain all living organisms. But what if we could unlock the entire sequence of human DNA, all 3 billion base pairs of it? Well, that's exactly what the Human Genome Project set out to do. In 1990, scientists embarked on a grand mission to decode the entire human genome, the complete set of genetic instructions. And the magnitude of this task was enormous. Imagine trying to sequence 3 billion base pairs at a cost of $9 billion and storing it all on the pages of books. You would need 3,300 books to store all the information from a single human cell. That's a colossal effort, but with advancements in genetic engineering and bioinformatics, the Human Genome Project was launched and completed in 2003, changing the future of biology forever.
So, why did scientists take on such an enormous project? What were the goals? The Human Genome Project had several clear objectives. First, scientists wanted to identify all the genes in human DNA. We're talking about 20,000 to 25,000 genes that control everything from eye color to susceptibility to diseases. Second, they aimed to sequence all 3 billion base pairs of human DNA to unlock the genetic code of life itself. But the impact goes beyond just sequencing. It was about storing this information in databases so researchers worldwide could access it, and developing better tools for analyzing DNA. And of course, there was an ethical component to this research, exploring how this knowledge could be used responsibly in medicine, healthcare, and society. Now, how exactly did scientists go about decoding the human genome? Let me take you through the exciting methods they used. The process started by breaking down DNA into smaller pieces because DNA is far too long to be sequenced all at once. These fragments were cloned in bacteria and yeast, creating what are known as artificial chromosomes. Then, automated DNA sequencers working on a method developed by the brilliant Frederick Sanger read the DNA sequences. But here's the tricky part: the fragments overlapped, and putting them back together was like solving a massive jigsaw puzzle. So, bioinformatics, a field combining biology and computer science, was crucial. Specialized computer programs helped align the overlapping fragments, resulting in the first draft of the human genome. After all that hard work, what did we learn? The human genome is made up of about 3.2 billion base pairs. But here's the surprising part: only about 1.5% of our genome actually codes for proteins, the building blocks of our bodies. The rest? Well, it's still a mystery. Interestingly, humans have only 20,000 to 25,000 genes. That's far fewer than previously thought. But don't be misled, because the functions of over half of these genes are still unknown. And here's an amazing fact: 99.9% of our genetic code is the same in every human on Earth. That tiny 0.1%? That's where all our individual differences, like eye color, height, and even disease susceptibility, come from. Now, let's talk about something fascinating in our genome: repetitive sequences. These are stretches of DNA that are repeated many times, sometimes even thousands of times. But guess what? They don't code for proteins. Scientists used to think these repetitive sequences had no purpose, but we now know they play important roles in the structure and function of chromosomes. They might even give us clues about how our chromosomes evolve and change over time. In fact, some repetitive sequences are used in DNA fingerprinting, helping us identify individuals based on their genetic makeup. Why is the Human Genome Project so important? Well, it opened up an entire field of personalized medicine. Imagine being able to predict which diseases you might be susceptible to or which treatments will work best for you, all based on your genetic makeup. But the HGP didn't just help us understand humans. It also shed light on the genomes of other organisms like bacteria, yeast, and even plants. This could revolutionize everything from healthcare to agriculture to environmental science. However, the work doesn't stop here. The real challenge now is to understand the functions of all those genes and how they interact to keep our bodies functioning. And as research progresses, we will continue to unlock more secrets about the vast complexity of life itself. So, what have we learned today? The Human Genome Project is one of the most groundbreaking scientific endeavors in history. It has mapped out the DNA that makes us who we are and laid the foundation for countless advances in medicine, biotechnology, and more. But this is just the beginning. The future of genomics holds incredible promise, from curing genetic diseases to creating sustainable solutions for agriculture and beyond. The possibilities are endless, and as we continue to explore the human genome, we'll uncover even more about the fascinating blueprint of life itself. I hope this session opened your eyes to the incredible world of genetics. Until next time, remember, biology truly is the science of life, from the tiniest cell to the complexity of the entire organism.